Chapter 2, Section 11, chemotaxis. Taxis is basically a directed movement, okay? All prokaryotes, all single-cell eukaryotes are moving, okay? Especially if they're in some form of a water or liquid column, okay? They're floating around. Brownian movement means they are kind of just tumbling. They're not stuck. Boom. Not to move, ever. So there is movement. Taxis is different. This is a directed movement in response to something, in response to some form of a stimulus. Whether it's chemical, chemotaxis, there's going to be some microorganisms that are drawn to light, phototaxis. There's the magnetic one that we've talked about in the previous sections and things like that for the magnetic fields. So this chemotaxis, phototaxis, whatever, it's either going to be a positive or negative, drawn towards something, or it's a signal to be elsewhere. We find this in both our bacteria and archaea. We also find these responses, but using different mechanisms, in protists, fungi, and whatnot. We study it most often in E. coli, okay? Peritricheous flagellated bacteria, okay? So talking chemotaxis. Turns out that they're on the surface of the bacteria, the outer membrane, they're in chemoreceptors, the cytoplasmic membrane. There are chemoreceptors. These receptors bind to certain chemical compounds. Certain chemical compounds, such as sugars, that's a positive chemotaxis stimulus. Other ones, could be negative. Time to move away, time to be elsewhere. Turns out that it's not a one and done. It's not just a perfect switch. Okay, there's one receptor triggered. I need to swim towards something. I need to swim away from something. No, it's going to be more of a threshold event. These concentration gradients, because remember, everything diffuses through a liquid. So these microbes are going to respond to different concentrations in their environment. Low concentration over here, high concentration right here. So if it's sugars, it's going to want to somehow get over to right there. If it's low concentration, high concentration of something that's bad, it's going to want to move away and be somewhere in this area. So that's why it's, temporal, not spatial. Okay, they're not choosing I want to be over here, not over there. They call this run and tumble. The run and tumble, if there's no attractant present, it's just randomly moving. Random runs as the flagella pull it along, flagella stop, it tumbles, moves around in three-dimensional space in the liquid, and then kicks in again. Boom, runs, tumbles, runs, tumbles, runs, tumbles, and it'll just keep doing this. And it's staying in the same general region, unless there's an attractant. Run, tumble, run, tumble. And notice it's not going directly from low to high concentration. It's using this law of averages that, random direction, take off, random direction, take off, random direction, random, until it hits the maximum, whatever this attractant was. Law of averages says that at some point in time, these will all add up and it will end up over here. It's just going to take time. It could be just a quick 1 or 2 tumbles and hey, I'm there, or it could be a couple of dozen before it's up here at the high end. But it will occur given time. When it comes to the polar ones, such as Pseudomonas or, Rhodobacter, Pseudomonas doesn't use the so much tumble as often. It just goes on runs. Stops, runs, stops, runs, stops. We can actually measure this chemotaxis. We have mechanisms for actually watching this, not so much in real time, but we can see the effects given enough time, going back 5 minutes, 30 minutes, an hour, 2 hours. We can watch these things move. Move towards stuff, move away from stuff. Whether we're using some form of attractant or something that's going to trigger the runaway, a repellent.